The omega-3 paradox. How fish oil supplements might impact brain recovery. For decades, fish oil supplements have been marketed as a brain booster, widely praised for their neuroprotective qualities. However, new research is challenging this blanket assumption, suggesting that one specific component of these supplements, EPA, eicosapentaenoic acid, may actually hinder the brain's ability to repair itself following an injury. The study, Memory Loss and Metabolic Shifts, researchers at the Medical University of South Carolina, MUSC, conducted a study using mice to observe how different omega-3 fatty acids affect recovery from mild traumatic head injuries. The results were unexpected. Impaired learning. Mice fed diets high in EPA performed significantly worse on spatial memory and learning tasks following a head injury compared to those without the supplement. Vascular interference. Instead of aiding recovery, EPA appeared to interfere with the repair of blood vessels by reprogramming their metabolic activity. The metabolic vulnerability. Researchers termed this phenomenon a context-dependent metabolic vulnerability. Essentially, under the stress of an injury, EPA may cause cells to shift their energy usage in a way that distracts from the critical work of repairing brain tissue. DHA versus EPA. Not all omega-3s are equal. A crucial takeaway from the study is that these negative effects are not universal to all omega-3s. The researchers found a distinct difference between the two primary types found in fish oil. DHA, docosahexaenoic acid. Known for its role in building and maintaining brain cell membranes, DHA did not interfere with the repair processes. Follow-up experiments involving human-derived brain cells. EPA, eicosapentaenoic acid. This fatty acid was found to accumulate in the brains of the mice, but rather than protecting them, it appeared to destabilize blood vessels. This distinction is vital because it highlights that omega-3 is not a monolithic category. Different fatty acids serve vastly different functions in the nervous system. Connections to chronic brain degeneration. The study's findings raise serious questions about long-term brain health, particularly regarding chronic traumatic encephalopathy, CTE, degenerative disease linked to repeated head trauma. In the mouse models, the blood vessel instability caused by EPA was linked to the buildup of toxic tau proteins, a hallmark of brain degeneration. When researchers analyzed human brain tissue from individuals with CTE, they observed similar patterns of metabolic disruption and blood vessel damage. This suggests a potential, though unproven, risk. If EPA impairs the cellular recovery process after a mild concussion, it could inadvertently exacerbate the damage that leads to long-term neurodegenerative conditions. The shift toward precision nutrition. It is important to note that this research is currently based on animal and cell models. While these findings flag a significant association, they do not yet provide a definitive warning for human consumption. However, they do signal a necessary shift in how we view supplementation. As neuroscientist Honor Eskiakek notes, the idea that fish oil is a one-size-fits-all benefit is increasingly outdated. The scientific community is now moving toward the concept of precision nutrition. The idea that nutritional interventions must be tailored to the specific biological context and the existing health of the brain. Conclusion: while fish oil remains a widely used supplement, this study reveals that EPA may have unintended consequences for brain repair and vascular health following injury. Future clinical trials will be essential to determine how these complex interactions play out in humans and how to optimize omega-3 intake for brain resilience.